The theory of panspermia suggests that life may span the universe, spread by celestial bodies like comets, asteroids, and even human-made spacecraft that unintentionally carry microorganisms with them. While it doesn't address the question of life's origins, merely pushing it back on other cosmic bodies, this theory speculates that forms of life capable of withstanding outer space could be transported by debris following celestial collisions. Its focus is less on how life began, but rather on ways it may have been disseminated through the cosmos. Pseudo-panspermia argues that the foundational organic elements for life were born in space, steadily spread to budding planets, and eventually fostered the development of life. This latter belief holds that these organic molecules, potentially even life, came from outer space and was distributed on Earth. Ever since Anaxagoras wrote about this theory in 5th century BCE, panspermia has been explored in varying degrees of scientific detail, as seen in the works of notable figures like Berzelius, Richter, Kelvin, Helmholtz, and Arrhenius. More recently, proponents such as Fred Hoyle and Chandra Wickramasinghe suggested that interstellar dust mostly composed of organic carbon, and these life forms keep entering Earth's atmosphere, potentially causing epidemics, new diseases, and even promoting macroevolution. Panspermia can take place either between different star systems, interstellar, or within a single star system, interplanetary. Various methods of such cosmic dispersal have been explored, such as radiopanspermia, propulsion of microbes through radiation pressure, lithopanspermia, transfer of organisms inside rocks, and directed panspermia, deliberate seeding of planetary systems with life. There are precautions to prevent contamination from space probes, but some organisms might not be eradicated entirely during the spacecraft assembly cleaning process. Even though it's still unverified, the possibility of life existing beyond Earth remains a persuasive scientific conjecture. The 1903 theory by Svante Arrhenius, known as radiopanspermia, suggested that tiny life forms can traverse space, with their movement powered by the radiation present in stars. According to Arrhenius, miniature particles of no more than 1.5 would be propelled through space at high speeds due to the radiation force emitted by the sun. However, for larger particles, this phenomenon is not valid, as the gravitational pull of the sun would overpower the radiation force. Critiques of the radiopanspermia theory emerged from leaders in the scientific community, namely Iosif Shklovsky and Carl Sagan, who highlighted that space radiation could be deadly to these tiny life forms. In a subsequent study conducted by Wallace and Wickramasinghe in 2004, they argued that bacteria or groups of bacteria had greater likelihood of surviving despite the lethal conditions of space. Experimental data from multiple satellite missions indicated that only bacteria embedded in substances such as clay or meteorite dust and protected from solar UV could potentially survive in harsh space conditions for a significant period. However, even while shielded, the spores would need additional protection from cosmic radiation that could be harmful to their DNA. As such, the transport of unprotected DNA or RNA during flights that rely solely on light pressure is highly unlikely, putting a damper on the potential validity of radiopanspermia. The possibility of transporting more substantial shielded spores into outer space, perhaps through comets' gravitational capture, remains unexplored. For the survival of microorganisms capable of enduring cosmic radiation, rocks at least 1 meter in diameter would be required for effective shielding. This stipulation discourages the likeliness of the radiopanspermia theory. Alternatively, lithopanspermia proposes that organisms can travel from one planet to another via the movement of rocks in either interplanetary or interstellar space, such as asteroids or comets. This concept, while still speculative, suggests organisms could even cross star systems on wandering planets or moons. Such a journey, while taking millions of years, is still feasible in terms of the billions of years life on Earth has evolved. Though there is no direct evidence supporting lithopanspermia within our solar system, this idea is becoming more testable with advancements in experimental science. The primary theme of the investigation centers around the exploration of potential survival of microorganisms enduring rapid Earth's atmospheric entry, termed as hypervelocity. The experiment involved bacterial spores transporting to approximately 120 km height via a two-stage Orion rocket. Microbes merged onto granite domes twice undertook these hypervelocity atmospheric trips. The results revealed that the spores could live in the rock's circumferential sections, being unable to withstand the forward-facing surface with a high temperature of 145 degrees. These results indicate that atmospheric transition could serve as a barrier for organisms like photosynthetic ones, which necessitate light availability near the rock surface. Albeit cyanobacteria's ability to endure unfavorable conditions such as desiccation and freezing, the stone analysis revealed that these organisms could not survive atmospheric entry. However, deep-lodged small non-photosynthetic organisms within the rocks might prevail through both exit and entry processes, including impact survival. The concept of directed panspermia was also explored. This suggests the intentional transfer of microorganisms in space, either initiating life on Earth or seeding new life on Earth or seeding new life on other planets through introduced microorganisms on lifeless planets. Legendary individuals like Francis Crick and Leslie Orgel conjectured life might have propagated intentionally by a highly developed extraterrestrial civilization. However, Crick later speculated that life could have originated on Earth. An alternative hypothesis called cosmic garbage, trialed by astronomer Thomas Gold in 1960, proposed that terrestrial life may have inadvertently originated from a trash pile abandoned by extraterrestrial beings. Directed panspermia could theoretically be proven by spotting an intentional signature message imbued into the genome or genetic code of initial microorganisms by our hypothetical progenitor nearly 4 billion years ago. One idea proposed that the bacteriophage Faxone 174 might be the representative of such a message. However, no known mechanism could prevent mutation and natural selection eradicating such messages over extensive time lapses. An independent fragment of the Orgwell meteorite found in 1965 created significant excitement. Kept in a sealed jar since its discovery, the fragment showed an embedded seed capsule while maintaining an undisturbed glassy outer layer. However, the unveiled seed belonged to a European Juncaceae or rush plant, which was artificially inserted and camouflaged using coal dust with the outer fusion layer being glue. The unidentified hoaxer apparently aimed to sway the 19th century debate on spontaneous generation, not panspermia, demonstrating the transformation from inorganic to biological matter.